If you if you allow us, we are going to record the session right now in order also to send to other companies that mentioned me that they couldn't attend today, but they want to see the report on the presentation that you will do today. So welcome to this session with part of the Ecuadorian banana cluster. Thank you again, Doctor.
Well, thank you very much, Jose, and thank you for the invitation. And good morning from Australia. So it's a wet, cloudy morning where I am in Queensland. So it's good to be inside. Okay, let me start by saying certainly thank you for the invitation to present today. I put together a presentation which probably is the best way to start. And very happy, obviously, to answer any questions after that. So let me just, yeah, okay. And go to there. And I've got to just trying to find where that just bear with me for a moment while I
You are able to share.
I'm trying to share now and just having a little problem. I might just have to shut down the presentation and reopen it. If you just bear with me for a moment. For whatever reason, it's not allowing me to share my PowerPoint.
If you go down to share screen and
yeah, got all that.
Yep.
Yeah.
You can send me the PowerPoints as well, Doctor, through email. And
that'd be great, but it's much too large. I'm just trying to figure out why it is not allowing me to share. Because it's giving me all sorts of options, but not the option to share. Let me just try again. It's there, but it's not sharing. Isn't that annoying? So
let me see.
here we are. Is it coming up yet?
No.
Dear idea, that's very annoying.
Probably if you send it to me by email or WhatsApp, doctor.
Yeah, I'm getting closer, I think. Um, Still not coming up. Wow, that is really strange because it's telling me I'm sharing it. Why is that just not coming up? That is crazy. Okay, so what I'm going to have to do, um, okay, Jose, I have just sent you the link. And I have no idea why it's not showing up on the screen.
You send it to me by email?
By email, yes.
All right.
Has it arrived?
Not yet.
So it's come?
Yes, it arrived. I'm right now downloading.
Oh, good. Excellent. I don't know why I can't share it. It's
Uh, asking for a verification code. Sorry, everyone. Okay, ready. Let me share the screen. Sorry, everyone. Doctor, can you see my screen?
And it's not coming up for me yet. Blank. Can you see the presentation? Ah, there we are.
Yes,
Yeah, that's it.
All right.
That's good. Okay. So if we want to go back to that.
Okay.
So Jose, will you change the slides or can I?
Sorry?
I think you will have to change the slides for me.
No, don't worry. Don't worry. I'll change it.
Okay. Well, apologies for that, everyone. I have no idea why my browser wouldn't allow me to share it. Okay. So this is an overview presentation. For me, I'm giving it on the 18th of April. You're seeing it on the 17th of April. Next slide, please. So I'm going to start with talking purely about Fusarium wilt or Panama disease tropical race 4, obviously commonly known as TR4. Next one, please. Okay. And you probably all know this very well, but it's always good just to start off with a description. So it's caused by a fungus, Fusarium oxysporum cubensi. This is a soil-borne fungus. It can survive for at least 50 years in the soil, maybe longer. Nobody really knows how long it can survive. It can live on other plants, particularly grasses and weeds, but only mildly. It reproduces asexually, so it doesn't recombine. That's probably one of the reasons why resistance seems to, in Cavendish, seems to have been maintained for such a long period of time. It's spread in, whoops, I just lost that slide. Okay. It's spread obviously in infected suckers and soil, whether that's on boots or vehicles or whatever, in irrigation water and runoff water and flood water. There's no chemical control that is effective for any period of time. And most importantly, this fungus kills Cavendish, but also many, many other cultivars. It's not only Cavendish. Next slide, please. Okay. It's been in Australia for quite a long time. We believe it probably evolved in Southeast Asia, particularly in Indonesia and Malaysia. It was introduced into Australia and into the Northern Territory in the late 1990s. So we've had quite a lot of experience with it. That plantation on the top left-hand side is the plantation we work on. It's the last commercial plantation in the Northern Territory in Australia. That plantation is probably about 18 months old. And you'll see there are already lots of yellow looking plants and gaps in the the plantation. So this fungus moves quickly. The way that this plantation works in the Northern Territory is that they plant and harvest the plant crop and the first return, and then they plough that back in and plant a new plot of land. So constantly replanting, and that's the only way they can produce bananas in this infected soil. And you'll see there the typical symptoms. Bottom left, the yellowing leaves, and then when you cut that shooter stem open, the very, very severe vascular browning or vascular necrosis, and it has a very, very distinctive smell. So once you've smelt it, you won't forget. So it's a very strong smell. Next slide, please. This is a photo I took of the Philippines. This is now 10 years old. The Philippines very, very severely affected, and one of the ways that they're controlling, well, not controlling, but managing to continue to produce is to open up new land. But they also have been attempting to, when they find a diseased plant, to pull it out and burn that area. So they'll burn all the affected plant material and also put a fire over the top of the infected mat. How effective that is, we don't really know. Next slide, please. So as I said, it probably started in Indonesia and Malaysia. So that's one of the centers of origin of bananas. And you'll see there in red, I've highlighted Indonesia and Malaysia. It's probably been there for probably thousands of years, co-evolving with bananas in the same area. Next slide, please. Then between, whoops, you went too far. Just yeah, that's good. Then between 1960 and 2000, it was then found in Taiwan. It was then found in Australia, in the Northern Territory, I said it earlier, but also in China. So it was really starting to move, but then it really got going. Next slide, please. So between 2000 and 2015, it really started to move significantly. So it moved into the Philippines. So that was the first of the banana exporting countries that became infected. It's moved into the Middle East, in Oman and Jordan. It was then found also in Pakistan, South Asia, and importantly, moved into Mozambique. And almost certainly it was taken from Philippines into Mozambique, where they were setting up new plantations in Mozambique to develop an export market. Next slide, please. Then really got going after that, moving into particularly India, into Israel, and Myanmar, Laos, Vietnam, and also into the UK, which was an odd one. So you're now starting to see that whole Southeast Asian banana growing region being highly infected. Next slide, please. And then, of course, the area that is really of greatest concern to you in South America and in Ecuador. So in 2019, it was found in Colombia. It's now been found in Peru and in Venezuela. And also, when we go back to Southeast Asia, it's been found in Cambodia and Thailand. So it's probably right across all of that region now and continuing to move. So when you see that slide, you realise that this is a fungus that's moving around very effectively. Unfortunately, it's humans that move it around rather than anything else, moving it around large geographical areas, and then locally it can move obviously in infected irrigation water or floods, etc. Next slide, please. So we set out quite a long time ago, that was around about nearly 25 years ago, to develop genetically modified Cavendish with resistance to TR4, tropical race 4. The reason we did this was we developed the technology to genetically modify Cavendish, and at that stage in Australia, we realised that TR4 was going to be a significant problem in the future. Next slide, please. So we were able to access seedlings from a wild seeded banana called Musa acuminata malacensis. It's one of the original parents of Cavendish, and it grows naturally in Indonesia and in Malaysia. But the most interesting part of that is that many of its versions are completely immune to TR4. So we were able to access seedlings, because this is a seeded diploid, seedlings which were segregating for resistance. So we had accession 850 and accession 848. So accession 850 was highly resistant or close to immune to TR4, whereas accession 848 was highly susceptible. So that gave us the opportunity to say, okay, this plant has resistance genes to TR4, and if we can access those genes, or if we can find that gene, then this is going to be really useful. Next slide, please. So we used a range of analyses, particularly genomic analysis and expression analysis in the resistant versus susceptible seedlings, to try to identify a resistance gene, and we came up with an extremely good candidate, which we've called RGA2. Now, so we cloned that resistance gene out of malacensis, and we transferred it into Cavendish by genetic modification. And then we planted our first field trial near Darwin, so that's in the Northern Territory, in January 2012. Next slide, please. Okay, so this is essentially what we've done. We've taken this gene, it's a banana gene, RGA2, from Musa acuminata malacensis, and we transferred it into Cavendish, and the Cavendish that we used initially was Grandnain. Next slide, please. So in 2012, we took 7 lines up to our field trial site in the Northern Territory, and that small-scale field trial was planted in 2012. Next slide, please. So this was a large commercial plantation. It had been severely affected by TR4 for more than 10 years, and we planted our field trial adjacent to commercial plantings, but it had been a field that had already been wiped out with TR4. We kept a complete history of every plant in the trial, and that's given us really important data about the rate of infection, the spread, recovery, etc. Next slide, please. So these are the results. This is after 3 years in that field trial site in the Northern Territory. In blue, the blue bars are the controls, and the red bars are the genetically modified lines with the RGA2 gene. So when you look at those, and as I said, this is after 3 years. When you look at those blue bars, the first one is GCTCV218. That's the other name for Formosana. That's the selection that comes out of Taiwan. The next blue bar, and that had close to 70% infection after 3 years. Williams was around about the same. It had about 66%. DPM25 was a mutant that came out of a Queensland program, which was supposed to have resistance to subtropical race 4, but it didn't have any resistance to tropical race 4. And you see it's got 100% infection, and there's Grand Name next to that, and it had around about 85, 86%. Then you look at the red bars, and 4 of those lines had significant levels of resistance, and particularly one we called RGA23, which had no plants. But this was a small field trial. And so that gave us tremendous hope that this gene really was doing something very, very significant. To get 4 out of 7 lines, I haven't got the 7th one there, 4 out of 7 lines with significant levels of resistance compared to the controls was absolutely amazing. Next slide, please. Okay, and this is tracking that resistance over the 3 years. So the lines in green or yellow are the genetically modified lines, and the lines in red are the controls. So there are a few important things that came out of this trial very early on, and that is that it actually takes quite a long time for those first plants to become infected in the controls. And you can see sometimes we didn't get any of the controls becoming infected for the first 12 months. So often people have very short field trials, but that's of no use at all. And you'll see, and one of the ones that's really important is one of the green lines, RGA24, which tracked along the bottom with no plants infected until right at the end of the trial. Next slide, please. And this is an analytical slide showing that the RGA2 gene actually occurs in Cavendish. That's really what that slide demonstrates. And then if we go to the next slide, this is what we know as the expression of the resistance gene. And you'll see those 4 lines there, RGA22, 23, 24, and 25, have significant levels of expression of RGA2, whereas right on the left-hand side is Grand Name, which is highly susceptible. And while it does have the gene, it just doesn't work very much at all. So that's probably the reason why Grand Name is susceptible, and these resistant lines we've selected out are so good, they have high levels of expression of RGA2. Next slide, please. And so that's what it looked like at the end of the trial. This is on the day that we actually had to pull out the entire trial. On the left-hand side, there is a non-transgenic control. It was one of the few plants that was still left with that very severe vascular necrosis, RGA23 in the middle. And then we did try another gene, SED9, and which also gave resistance, but it's a gene from a nematode, so we haven't progressed with that at all. But the RGA23 and also RGA24 lines looked absolutely fabulous. When you cut them open, there was no vascular necrosis at all. Next slide, please. Okay, so we've published that. That was published quite a long time ago. One of the important things we like to do is to publish our work as we go. And so that was published in a journal called Nature Communications, and that's easy to access if anyone would like to read all of the significant details of that trial. Next slide, please. Okay, so that was a small-scale field trial. We really needed to understand how those lines would perform in a large field trial, and we planted that field trial in 2018, and it's still going. So next slide, please. Okay, so this is what this field trial looks like. This is when we This is probably around about the time of the harvest of the first plant crop. So big trial with all 4 resistant lines and also all the necessary controls. So we had Grand Nain and Williams in there as well. We didn't bother putting Formisana in because it was so susceptible in the first trial. Next slide, please. So I said this is a large-scale field trial over now 5+ years. It's going on 6 years now. It was designed very much to mimic a commercial plantation, so it's managed the same way. All the plants were taken up to Darwin Fruit Farm and acclimatised 3 months before they were planted. And so we took those 4 original Grand Nain RGO2 lines into the field plus controls. And importantly, measured many of the important agronomic characteristics. And we're just writing that those results up now to be published. Next slide, please. Okay, so this is after 3 years. And the names have now changed from RGA2 to QCAV. We needed a more definitive name for registration. So after 3 years, and as I said, this is a large field trial, you'll see that Grand Nain had an infection rate of around about 58%. Williams was worse, it was at 68%. And the best of the lines was QCAV-4. So after 3 years, we didn't have any plants infected from that line. The other 3 lines still gave good levels of resistance. QCAV-3 was 6%, QCAV-5 with 10%, and QCAV-2 with 18%. So still significantly lower than the controls. But QCAV-4 really was a standout line in this trial after 3 years. Next slide, please. And this again shows that rate of infection over, this is over 6 crops. So this is plant crop, first, second, third, fourth, fifth, and 5th return. What we're now finding now is after the 5th return, we're getting a couple of plants of QCAV-4 infected, not many at all, but a couple. The interesting thing is that when they get infected, the sucker, when it comes away, appears to be completely healthy. So where the controls, and you'll see after those 6 cycles, Williams is close to 100% infection, and Grand Nain is around about 75% infection, and QCAV-4 way down the bottom there with around about 2% infection at that stage. Next slide, please. This was very important. So we measured bunch weight over those 5 cycles, and these are the bunch weights for when we say Williams and Grand Nain only from healthy plants. So the equivalent here is QCAV-4 and Grand Nain, because QCAV-4 background is Grand Nain, and you'll see there that it's very, very close to the bunch weight of QCAV-4 is very, very close to the bunch weight of the Grand Nain control. The other important thing is that what we found is QCAV-4 had a slightly shorter cycle time, and so the yield per hectare per year was slightly better with QCAV-4 than Grand Nain. So that's tremendously important because this isn't a line that is being affected and we're not seeing any yield reduction at all, which is wonderful. Next slide, please. Okay, so this is what's happened, and this is yield in tonnes per hectare over 5 cycles. as this is what happens in a real situation. You'll see that QCAV-4 is tracking around about 40, continuing to track around about 40 tonnes per hectare, whereas Grand Nain, because so many plants are being lost, is really starting to decline, and at that stage is below 20 tonnes per hectare. And if we measure it now, it'd be, I don't know, probably about 5 tonnes per hectare, because the level of infection has just continued to go up with Grand Nain, whereas QCAV-4 is very stable. Next slide, please. So really, after those 6 cycles, the 6 crops, the plant crop plus 5 returns, with QCAV-4, we only lost 2 harvests out of 300, which was fabulous. Bunch weight, very close to Grand Nain control, cycle time slightly shorter, and QCAV-4 was transformed with a single banana gene. And this gene occurs in virtually all domestic bananas, but in the vast majority of those, it just doesn't work. It's switched off. Next slide, please. So we now have Cavendish bananas with close to immunity to TR4. It's a reality, with no impact on yield. So I think one of the most important things is, there have been so many comments that, if this is the end of Cavendish. No, it's not. Whatever happens, we will have a Cavendish banana that is highly resistant to tropical race 4, and that'll always be available. Next slide, please. Okay, so what we've done now, just over 12 months ago, because this is a genetically modified banana, they're highly regulated in Australia and anywhere else in the world. So we had to apply for deregulation in Australia. This was the first genetically modified banana submitted for deregulation anywhere in the world. We had to apply to 2 groups, the Office of the Gene Technology Regulator, which regulates production, and Food Standards Australia and New Zealand, which regulates consumption. Next slide, please. So in February, approval for the commercial production of QCAV-4 was given in Australia. So this is the first genetically modified banana released anywhere in the world, and it has real disease resistance. So I think it makes a very powerful new technology available to banana growers. Next slide, please. Okay, so that's the genetic modification story. That's been a highly successful research and development program going into a commercial program now. But as many of you know, not everywhere has the same attitude to genetic modification. So we know that in Europe, there are very, very few genetically modified crops being grown. Most people believe there are none. That's not true, actually. There are places that are growing, European countries which are growing genetically modified maize, and they import massive amounts of soybeans and corn, maize, into Europe particularly as an animal feed. So it's not that Europe is not a place you can send GM crops. You can, but it's just extremely difficult. Next slide, please. So one of the ways to go, and I'll put here, what are the pathways to new banana cultivars? So many of you would be aware of the somoclonal variants of Cavendish, and Formosan is one of those. There's another one called Nova Green. There are a number of them. And yes, they show tolerance to TR4, but it's virtually impossible to ever develop those with Sigatoka resistance or any other future diseases, because these are random events. It's not easy. You're not going to try to improve them through specific genes. So these are random events. So if you then look at genetic modification, again, the cultivar that you're looking at is Cavendish, and the only real example of that is QCAV-4, which has high resistance to tropical race 4. Can we put in Sigatoka resistance? Yes, almost certainly we can. And if there are future diseases, we can probably do that as well. Gene editing, which is a different process. So the difference between genetic modification and gene editing, in gene editing, we're only altering the genes that already exist in Cavendish rather than putting new genes in. So with gene editing, we can work with Cavendish, obviously, and there are no practical examples of that yet. It's in progress. So certainly us and a number of groups, or a couple of other groups, are looking at both. Diseases would also be possible. So if we then look at conventional breeding, and there are a number of programs, as you know, FOCR is probably the most advanced, but also Embrapa in Brazil and Yellowway in the Netherlands. So there's a number of programs. What will come out of that?
Hello, doctor.
Next slide, Jose.
All right. Sorry, I think your signal came. Hi, doctor.
That's okay. So the advantages of gene editing are that they're considered or are under consideration as being non-GM in most important markets. So that's particularly in North America, many of the South American countries already, Japan, the UK, and also in Europe, it's under consideration as non-GM. So that's a really big change. It hasn't been fully approved in Europe, and it'll probably take a number of years for that to happen, but it's certainly on the way. So we can make small changes to virtually all the highly accepted cultivars, and that includes Cavendish. And also, you can use continuous improvement, as you can with genetic modification, but you can do that by adding new traits when required, such as a new disease. Next slide, please. Okay, this just gives you an idea of where all these different techniques fit. At the top there, we've got breeding, natural mutations. They're the sort of somoclonal variants, and mutagenesis, and there are some examples of that in bananas as well. The next group are the gene editing group. And I won't go into the sort of technical details, but SDN1 on the top left-hand side there is the technology that we use, and it's the one that is most likely to be approved for Europe, whereas SDN2 and SDN3 may not be. And right down the bottom there is transgenesis and cisgenesis, and yes, we're already doing those as well and continuing with that work. Next slide, please. Okay, so from GM to gene editing, I think what we call developing Europe acceptable TR4 and Sigatoka resistant bananas. So we're doing two things here. One is we're developing Cavendish with TR4 and Sigatoka resistance. But also we've taken another banana, which we call Goldfinger, but it's actually came out of the FOCR program, FOCR1, and it has high levels of resistance to TR4 and Sigatoka, but its fruit is not particularly acceptable to consumers. So we're using editing, and I'll show you a little bit of that in a moment, to edit the fruit of that. So not the resistance because it's already there, but the fruit to make it more acceptable. And there's a whole lot of really quite exciting things we can do about that. Next slide, please. Okay, so we're looking at diseases, particularly TR4 and black sigatoka. Obviously highly sustainable because we're getting away from the use of agrochemicals, and we are also looking at consumer traits and grower traits, and also looking at bringing back some of the old cultivars, and that's particularly Gros Michel. Next slide, please. So solutions obviously improve cultivar sustainability. We can develop these products much faster, and if we can develop Sigatoka resistance, obviously with a lot lower production costs. Next slide, please. So we've now got, as well as our genetically modified banana program, which we are continuing with because we're expanding from not only grand name but into Williams Cavendish as well. But for our gene edited program, we're developing non-GM disease resistant Cavendish. We're enhancing fruit quality in both Cavendish and in Goldfinger. And we're looking at bringing back old cultivars. And the major one we're interested in is Gros Michel, which you know is susceptible to race 1 as well as TR4. And it's a bit too big. So we're looking at reducing its height as well. Next slide, please. Okay, so the benefits of the gene editing program is that they will be treated as non-GM bananas in many jurisdictions. And that's important because not only getting market access, but also they won't be labelled and they don't require deregulation, which is quite an extensive process. So obviously access to major markets, but it's very much evidence-based disease resistance, and we can tailor that to particular cultivars. Next slide, please. So we've already developed non-GM gene-edited Cavendish, and they've gone right through to field trial. Next slide, please. So this is how it's done. We use a technology called CRISPR-Cas9 up on the top there. And these are essentially molecular scissors that we can go into a banana, into just one part of one gene and slightly alter that. And so we've developed that technology for Cavendish bananas, and it works very well. And we end up with edited, and we're using both Williams and grand name Cavendish. Next slide, please. So this is the process, and we use this process not only for editing, but also for genetic modification. And we start off with the immature male flowers. We generate what's known as a banana embryogenic cell suspension. This particular one is Williams. We transiently edit those cell suspensions, and then we select out those cells, and they've got to be individual cells. Those individual, and then from that generation and all the way through to plant establishment and maturation. Next slide, please. And this is an example of taking one of those plants, a single cell. So the single cell on the left-hand side has already developed into an embryo. We germinate that embryo. That's just close-up views. And then we can put the roots on those plantlets and take them through into the glass house. And you see on the bottom there 4 different examples of edited Cavendish plants that have been regenerated. Next slide, please. Okay, so this is the flow diagram. So we start on the left-hand side, those embryogenic cell suspensions. We've developed a non-GM gene editing protocol. Then on the right-hand side, we use genomic and transcriptomic analysis to identify susceptibility genes that we want to knock out and resistance genes that we want to improve. And then we then design and generate these editing cassettes, and then do the edits and regenerate and characterize those edited plantlets, and then take them through to the field for TR4 and Sigatoka infection. So one of the important things we do is we've learnt from experience, small plant tests in the glasshouse tend not to be particularly informative. We like to take everything through to the field to really understand the level of resistance that we get in a real situation. Next slide, please. So this is where we are with the timelines, and this is our program 1. This is our disease-resistant Cavendish. So we finished all of the edits. We finished those earlier this year. We've planted the first field trial, and so we're timing this through 3 field trials, 2 field trials here, which will be 12 months apart planting. And then we will be harvesting and identifying elite lines in the field, probably around about mid-2026 to 2027. So they're well along the way, and those lines we expect to be distributed around about December 2028. That sounds like a long way away, but I can assure you it isn't. It'll come around very quickly, unfortunately. Next slide, please. So for TR4 resistance, and this is also Sigatoka resistance, we've identified a panel of 24 genes. A certain amount of that work has come out from our QCAV-4 work, where we've identified which genes have been up and down regulated. So we've identified those genes, we've done the edits, and all of those edits are now done, and we're now progressing the lines through to the field. So the first ones are up in the field already, and the rest will be planted over the next 12 months. Next slide, please. So this just gives you an idea that we take different lines up. So we've got the first 6 genes, and all of those are up in the field now, and we've knocked out various parts of those genes to see whether by knocking them out we can generate resistance. Next slide, please. So that's just those plants up in the field in the screen house that was them being acclimatized before they were planted out. Next slide, please. So these are the other two programs that we've got: the enhanced fruit quality, the first field trials of the enhanced fruit quality will be planted in the field probably in mid-2025, and we'll be getting results from those pretty quickly. We've got some interesting results, and I'll show you those in the glasshouse already. The old cultivar program is a bit slower because for disease resistance, we really have to identify what genes to edit from program 1. So program 3 is a bit slower, and probably won't have plants in the field for another 2 years. Next slide, please. Okay, so let me talk a little bit about fruit enhancement in Cavendish and Goldfinger. So this has turned out to be a really, really interesting program because it's quite different to what most other people are doing. So we analyze the RNA and we analyze all the components, so all of the chemicals that are in banana fruit. And we do this with various technologies. So the idea was we took 4 cultivars, Cavendish, Goldfinger, Ladyfinger, which is a local banana in Australia, and also Ducasse, which is a small sweet banana, sometimes known as pisang awak. So we've taken those and we looked at 4 stages of ripening: that early green, late green, ripe, and full ripe. Next slide, please. So what we wanted to do is to identify what are the components of these different cultivars, and then identify how we can potentially improve them. So if you look at the sugar accumulation, which is really interesting, Cavendish has the most glucose and the most fructose. Not by a huge amount, but by a significant amount. The interesting one is Ducasse there, which is known as a small sweet banana, seems to have relatively small amount of sugars. And we're now starting to understand that and being able to utilize that. Next slide, please. Okay, so when you look at the acidity, and when we often talk about that acid sweet flavor, you'll see that here Ducasse has low acid levels, and so that's quite important. And so does Cavendish. Cavendish is, as we get to full ripe, the level of acid drops off significantly. So that's probably one of the reasons why we think Cavendish is really nice, because it's really quite sweet fruit. And so we started to understand what genes we're going to, or are editing. We've started those edits already to make really very, very interesting next generation bananas, both Cavendish and Goldfinger. Next slide, please. And the other interesting one is amyl acetate. So amyl acetate is the compound that smells like bananas. So if you take a vial of isoamyl acetate and smell it, you'd say, wow, that's bananas. So Cavendish has very, very high levels of isoamyl acetate. So that's the banana smell. That's what we're thinking. Wow, look, smell of Cavendish, and you're smelling isoamyl acetate. Whereas the other bananas have much less. So one of the things may be to increase the level of isoamyl acetate. Next slide, please. And here I've given you an example of a nutritionally enhanced Cavendish. So this is a standard Cavendish banana, which we've now altered to produce a particular compound. And it is probably the most spectacular looking banana. These deep purple leaves. And we're just analyzing the fruit is just forming at the moment. So we're expecting the flesh also to have this deep purple color. So these are the sorts of things we now have available to be able to really develop the next generation of bananas, commercial bananas. Next slide, please. And this is the flow chart for developing those fruit enhanced bananas, which we're using for both Goldfinger and Cavendish, and the Cavendish ones are coming along really well. And our targets are sweetness, sour, texture, aroma, and nutrition. Next slide, please. Just a quick one on our facilities. We've got a large tissue culture facility. Our laboratories are state of the art, and we've got both glasshouse and field trial facilities available, all within a relatively limited area. Next slide, please. So this is just some of the research team. I lead it. We've got, and some of you may have met some of these other people. Some of these have been to Ecuador. Next slide, please. So where are we going from here? So we've now established a company called Sustain Bananas, and it's different to other companies in that it's owned by and will be owned by people actually interested in the banana industry. So it's owned by QUT, that's the university where we do all our work. It's owned by researchers, and we're now attracting banana growers to become shareholders in Sustain Bananas to develop the next generation of Cavendish and other banana growers. And those banana grower investors will have very significant benefits, including early access to cultivars and reduced royalty rates and all sorts of really advantageous benefits. Next slide, please. Okay, so that's the end of my presentation. I'm very happy to take questions now, or I can be contacted either by phone or by email. And particularly, Jose, thank you for showing those slides. I do apologize. I have no idea why it didn't work from my end. So very happy to take any questions.
Thank you, Doctor, for the conference today. If there is any questions from the participants, please raise the hand or write it down within the chat. Doctor, and one question: what countries are you planning within this year to start the process of trial for this variety?
Yeah, so QCAV-4 is likely to be planted in the Philippines in the next 2 to 3 months. Just going through all of the approvals, because we have to send the plants from Australia up to the Philippines. They have to approve the field trials, etc. Now that's well along. The way we think the field trial approvals are just about complete, and then we'll be sending plants up and being able to screen them in the Philippines. So that's going to be really good to see how these plants perform in a different environment. But it's the same strain of tropical race 4 that we have.
And Latin American country, not yet this year? Or?
No, not yet. The number of countries, so we would be able to do it in Colombia because they accept GM, probably Costa Rica as well. So that a number of countries are possibilities, but we haven't progressed any of those as yet.
And is there a partner here in Latin America or per country, or is that directly through the University of Queensland or through a company, Doctor?
So we're in the Philippines. We'll be working with Del Monte. So they have an option over QCAV-4. I should mention QCAV-4 they have an option just on QCAV-4, and that's a Grand Nain type. We have new Grand Nain types with the same gene, the RGA2 gene coming through, but also Williams. So all of those lines are owned by QUT. So at some stage in the future, we'd be looking for partners, and we may well take those through sustained bananas as well.
Very clear. Okay. If there is any questions, if not, Doctor, I would like to also extend an invitation in our next events that we always celebrate here in Ecuador. You know, our convention in October. So we're going to reach out in a couple of weeks. It has been really interesting to hear the advances, the development, and all the trials, and we will be happy as well to start also being in contact if we decide within the cluster here and with all the affiliates to go through a process that will lead us to also be able to accept the GM variety. So we're going to be reaching out, Doctor.
Oh, I look forward to that, Jose.
Thank you.
And as I say, if you have any other questions, please contact me probably by email. It's the best.
Thank you. Thank you, Doctor.
Okay.
Have a nice day.
Thank you. Bye-bye.
Bye.